Welcome to this explainer. Today, we're diving straight into a fascinating ecological paradox. We're going to decode a recent scientific breakthrough about coral resilience and unpack this wild mystery surrounding what researchers are calling a microbial detox crew. Let's get right into it.
So we're starting with a real head-scratcher. How on earth do corals survive toxic pollution? We've all seen the grim headlines, right? Coral reefs dying off globally, mostly because of rising heat and severe coastal pollution. But in some heavily polluted areas like the western waters of Hong Kong, these corals are inexplicably thriving. They are surviving and even flourishing in what should technically be a highly lethal environment.
To really grasp the sheer scale of this mystery, you have to understand the normal limits. Usually, the nitrate threshold for coral survival is incredibly low, just 1 to 2 micromolar. Hit that level, and physiological processes literally begin to shut down. But in Hong Kong's hyper-eutrophic waters, these coral reef oases are flourishing despite huge spikes of up to 20 micromolar of nitrate. That is 10 to 20 times the lethal limit. It's an environment that by all accounts should be an absolute coral graveyard. Yet somehow, life finds a way.
Which brings us to part 1, the Hong Kong coral mystery.
You see, nitrate eutrophication, which largely comes from things like agricultural and industrial sewage runoff, acts like a massive poison to the coral ecosystem. It's really a multi-front attack. First, it fuels macroalgal overgrowth that literally smothers the reef. Worse than that, it disrupts the delicate symbiosis between the coral and its algae, effectively starving the coral host. It even restricts their biomineralization, meaning they can't build their calcium carbonate skeletons, and it induces severe oxidative stress. So under this intense multi-level biological assault, how in the world are the corals in Hong Kong fighting back?
Let's move to section 2 and meet the microbial detox crew.
Scientists looking at this incredible survival rate hypothesized that the corals were employing a metabolic detoxification mechanism. And at the heart of this mechanism is a process called denitrification. It's an enzymatic process where microbes literally reduce harmful nitrate or nitrite into harmless nitrous oxide or dinitrogen gas. Essentially, the working theory was that the corals might be using specific bacteria to simply gas off the toxic pollution right out of the surrounding water, kind of like their very own built-in water filtration system.
So to test this, the scientists mapped the coral's microbiome across both clean and polluted sites. Naturally, they expected to see a massive surge in the abundance of these detoxifying bacteria in the highly polluted western waters compared to the cleaner eastern ones. But here is the twist. When they looked at the dominant denitrifier genera, they found groups like Ruegeria at 12% and Pseudomonas at 12%, just completely evenly distributed across both clean and toxic reefs. The overall population sizes didn't change at all based on the pollution levels. No surge, nothing.
So what's really going on? Let's zoom in for section 3, the specialist twist.
The researchers were left with a true ecological paradox. As they put it, resilience is not mediated by diversity or abundance shifts in denitrifier genera. Basically, looking at the whole city of bacteria, the broad genus level, revealed absolutely zero clues to the coral's survival. The overall census of the bacterial city looked exactly the same whether the water was pristine or highly toxic.
Which naturally begs the question, if not the genus, then what? If the broad categories of bacteria weren't shifting to fight off this immense pollution, the scientists had to ask, who exactly inside that bacterial community is doing the heavy lifting?
To find out, they conducted a massive subgenus investigation. First, they went in and isolated 419 individual Ruegeria bacteria directly from the corals. Second, instead of just looking at their basic species tags, they mapped the recent gene flow of these isolates using some seriously advanced population genomics. And third, they used that data to delineate 29 highly specific subpopulations. By dropping down a taxonomic level and mapping the deep genetics of these individual isolates, they uncovered distinct, elite subpopulations hiding right under their noses.
And what they found was truly a game changer. They discovered a hidden team of denitrifying specialists, specifically clusters known as MC1, MC6, MC13, and MC18. Just think about the jump in numbers here. These elite clusters made up 1 to 10% of the community in the high nitrate, highly polluted western reefs, compared to just a tiny fraction, like 0.1 to 2% in the cleaner non-western reefs. They were massively enriched in the polluted waters. And fascinatingly, the researchers found many of these specialized bacteria were actually living deep within the coral skeleton itself, not just hanging out on the surface.
Let's head into section 4 and explore exactly how these specialists managed to survive.
The researchers didn't just stop at mapping the genetics, though. They actually tested their performance, and it revealed an incredible contrast in nitrogen gas production rates. In laboratory incubations specifically designed to mimic the low-oxygen environments found inside corals, these specialists reached up to 472 picomoles per milliliter per hour. They completely and utterly outclassed the regular, non-specialist bacterial populations in their ability to process these toxic chemicals.
To put that difference into a single, staggering perspective, think about the number 10. This elite subpopulation of specialists was literally 10 times more efficient at converting toxic nitrates into harmless nitrogen gas compared to their everyday non-specialist counterparts. They essentially act as supercharged detoxification engines running right inside the coral.
But how did they get this incredibly efficient? Well, they underwent intense genetic adaptations through convergent evolution. They actually depleted their active nitrogen transport systems, experienced a loss of assimilatory nitrate reductases, which is basically throwing away their normal nutrient foraging tools, and they even ditched their phosphorus scavenging genes. Why would they do that? Because they live in a nutrient-rich soup. They threw away their normal foraging genes so they could redirect all their metabolic energy into hyper-efficient waste removal. It is an absolutely amazing evolutionary trade-off.
Finally, Section 5, Rethinking the Coral Microbiome. The overarching takeaway here is just so vital for conservation. Conventional microbiome surveys miss ecosystem-scale adaptations entirely by only looking at the genus level. If marine biologists and conservationists only survey that broad genus level, looking at the whole city instead of the specialized individuals, we remain completely blind to the hyper-efficient microbial heroes that are actually saving the ecosystem from total collapse. So I want to leave you with a final, provocative thought. If corals can find microbial allies to survive our pollution, what other hidden lifelines are waiting to be discovered in a changing ocean? It really leaves us with a fascinating appreciation for nature's invisible capacity to adapt. It reminds us that sometimes you have to look incredibly close to see the bigger picture. Thank you for joining me on this explainer. Until next time, keep questioning and keep learning